Right, so let's get started. So welcome to the computational analysis of single-cell RNA-seq course, which is taught by myself. I'm Martin Hemberg, group leader at the Sanger Institute, which is about 10 miles south of here. And we also have a chap here with a very nice haircut, Vladimir, who is a staff scientist in my group. And we also have Tallulah, who's a postdoc, and we'll also have Davis McCarthy here in a little while, who is also involved in the course. So I think the important thing to tell you here is that we are currently taping this, or even broadcasting it live on YouTube, this lecture. So the camera sort of sits up there, and it takes most of me and the screen here. So if you don't want to be on the broadcast, make sure to stay away from this area. And other than that, we will be teaching. All the material that we'll be teaching are on this webpage. All the materials can be found on this webpage, which you should have open on your in your browsers. And if there's something that you've missed or want to go over again, you can just revisit it. And the second chapter, the technical requirements, contains instructions for how to install everything on your own machines. And then you should be able to run everything at home or at work just fine. And I think that's about it. Any questions at this stage? Okay, so let's get on. So the format of this course, the way it's going to work out, is that one of us is going to be talking a bit about a specific chapter here, and then interspersed throughout, and you should be sort of basically following along on your own computers. And then after a while, there will be some exercises, and the point is for you to work on those exercises, sort of either individually or together. And if you get stuck somewhere, you should raise your hand, and we will come and try to help you out. And then after we've given you sufficient amount of time to work on those exercises, we will try to round up and move on. And the overall idea is that we should be covering sort of all of this material. We hope to be done around lunchtime tomorrow, maybe a little bit later. And then what we'll be doing for tomorrow afternoon, if there is any time over, we will allow you to basically work on your own problems. So what you can do, we either have a bunch of what we refer to as advanced exercises, which are basically sort of open questions that you could, which we think are relevant to computational analysis, that you could sit around and work on. What people have done in the past is that they have brought their own data sets. And if you want to sort of try to carry out the type of analysis that you've worked on here through the course, apply that to your own data set, you can do that, and we'll be around to try to help you out. Or if you have anything else that you want to work on related to this topic, we will be here through the afternoon and help you with any questions that you may have. And of course, you're also welcome to head out for an early exit on Friday afternoon if you'd like to. Right, so first, before I talk about single-cell RNA-seq, we're talking about bulk RNA-seq, which I'm assuming that you're all familiar with now, because it's been around for almost 10 years. And it's a way of quantifying gene expression in a genome-wide manner. And it's safe to say that it has greatly transformed the field of genomics and the field of transcriptomics, and that it has allowed us to get a lot of interesting data and measurements that weren't accessible before. Perhaps the main disadvantage of bulk RNA-seq, which was known already at the time when it was published, is that in order to make it work, you need a large number of cells, typically on the order of hundreds of thousands or millions of cells, which means, of course, that you have what you end up seeing is an average of the whole population rather than the transcriptome of individual cells, which if your population is homogeneous, this shouldn't be a problem, but if there is a large degree of heterogeneity, you might be missing details by essentially smoothing them out and averaging across the whole population. So now there has been a lot of technological development, and actually already in 2009, the first paper where the authors were able to improve the protocol to such an extent that they could work with the RNA, which was obtained for only an individual cell. It was published in 2009, actually by the Surani lab here in Cambridge. But it took quite a lot of work, and it's only around 2014 when the method sort of started taking off. I would say that's a combination of improved protocols, so that it's just easier to carry out, and you get sort of more reliable results, but also significantly reduced sequencing costs, because it gets a lot more expensive to sequence single cells than to sequence bulk, just because of the sheer volume that you need to do. So now, of course, the main advantage of single cell over bulk RNA-seq is that you can look at what happens at the level of individual cells, which means that you have the sort of the ultimate resolution that we need for biology, and it allows us to exactly quantify how different individual cells are in a population, and we can actually start to delve into this heterogeneity. So as you're surely aware of, there's been a lot of interest and a lot of attention to this technology, and there's currently a huge amount of interest both on the technological side in terms of developing new and more efficient protocols, but also on the computational side in terms of developing new methods for analyzing all this data, as well as on the sort of more biological side in terms of figuring out how to use it to answer new questions in biology, but also on the clinical side in terms of how to figuring out how this can be used in medicine and other more translational applications. So, of course, here the focus will be on the computational side, and I think what happened initially was sort of parallel to what happened when the bulk RNA-seq was introduced about 10 years ago. So the first thing that people did when they tried to analyze the data was to use something that they were familiar with, which was microarrays. So people tried to use the methods that had been developed for microarrays and apply them to RNA-seq data. Of course, it works reasonably well, but not fantastic. And pretty soon there were methods that were specifically devoted to RNA-seq out and about, and they were doing a lot better. The same thing happened when single-cell RNA-seq was introduced. People started off by using what they had and what they were familiar with, and those methods work, but they're not fantastic. And it turns out that it's relatively easy to improve the performance by having methods that are specifically designed for single cell rather than for bulk RNA-seq data. So here, this is a picture taken from Wikipedia, which sort of basically illustrates the sort of the typical workflow for single cell RNA-seq. So it involves a few different steps. Turns out that what we believe, based on the data that we've seen, is that sort of the step of sort of dissociating the cells, if you take them from a tissue, is actually very important, because that can introduce a lot of stress to the cells and so on, and that can cause problems in terms of the transcriptome if it's not done, and the quality of the data if it's not done correctly. You need to isolate the cells, which can be done in a few different ways, and then after that we're basically back to the sort of standard RNA-seq workflow, where we need to extract the RNA, reverse transcribe it, turn it into cDNA, which can be amplified, and then churned through a sequencer. We get the data, and then once we map that data to a reference genome, and then we can quantify the amount of expression from each gene or transcript, and then we can start doing different types of analysis. And I think one of the important issues here is that the types of analysis that can be done here, for example, clustering, many of them are different from what you could do with bulk RNA-seq data, just because of the this high granularity of the single cell data. So we've sort of outlined here the typical workflow that you go through once you get your data. Sort of the starting point that we have in this course is once your data comes off the sequencing machine. So once you imagine that you're handed a bunch of FASTQ files, what do you do with them? So the boxes here are sort of colored. Consider this sort of the first 3 yellow boxes as steps that you would do for any kind of sequencing experiments, regardless of what it is. And so the methods that you use here do not need to be specific for single-cell RNA-seq, but you'd use probably the same types of methods for if you're analyzing DNA data or ChIP-seq data or anything else. The next few boxes are basically sort of quality control, and here we'll be talking about methods that have been specifically developed for single-cell RNA-seq on basically how to ensure that the data is of high quality. Whereas these yellow boxes are also quality control aspects, but again, they're at the level of reads, so they don't need to be specialized for this aspect. And then finally, the blue boxes here correspond to more sort of analysis question or sort of scientific or biological questions. So how can we carry out quantitative analysis to answer the scientific questions that we're really interested in? And we'll be discussing some of those towards the end of the course. So I think overall, when it comes to the QC, the main challenges that we're dealing with here in terms of the data is because we start out with such a small amounts of RNA, which means that the noise will be very high. So we're basically, in order to be able to get the results out, the RNA needs to be amplified. So you have very high level of amplification, which can, of course, result in large biases. And one specific feature of single-cell RNA-seq data, which is not present in bulk RNA-seq data, is this concept of dropouts, which is basically when, we'll be talking about that quite a lot here, but what happens is that you will have genes that, based on their average, appear to be relatively highly expressed, but still you'll see a substantial fraction of the cells have actually 0 reads detected for that gene. And that's what's referred to as a dropout. It causes all kinds of bother, including on the sort of pure algebraic level, because having zeros often makes life a lot more complicated, particularly if you want to sort of try to divide by them. So I'm going to talk very briefly about experimental methods. This is not what the course is about, but we figured that it's useful to give you a little bit of an orientation. So as you can see here, there is a large number. This is a non-exhaustive list of some of the protocols that I could think of that are out there today. And I think one important question is to sort of figure out which protocol is most suitable for you and your needs if you're going to be doing an experiment. And they can be sort of characterized in a few different ways. We've chosen to focus on 2 different aspects here, quantification and capture. So for quantification, we divide them into 2 types, either full-length or tag-based. So it's pretty much what it says on the tin. So the full-length protocols are the ones that try to quantify the whole RNA, the whole RNA. So whereas the tag-based protocols only try to get the 5' end or the 3' end of each transcript. So they have certain advantages and disadvantages. The main advantage of the full-length transcript is, of course, that you can look at what happens across the entire transcript, and which is very useful if you're interested in, for example, splicing or trying to quantify different isoforms. On the other hand, With the tag-based protocols, you can use something called UMIs, which stands for Unique Molecular Identifiers, which is something we'll be talking about quite a lot later on as well. The advantage of UMIs is that you can think of that as a little tag that you put on the, a unique tag that you attach to each transcript, and it significantly improves, or can significantly improve quantification. So that's why that might be quite nice. The other aspect that we're going to be talking about is capture. So primarily, there are sort of three main different methods that you can use, which we refer to as microwell, microfluidic, or droplet-based. And the main difference between them is really the amount of the kind of throughput. Microwell is typically the lowest throughput. Microfluidic platforms have slightly higher throughput, and droplet-based methods have really high throughput. And when I say really high, we're talking about you can easily get tens, hundreds, thousands, or even millions of cells in one experiment. So really what's holding you back there is your budget. In addition to the throughput, there are of course other aspects to consider. I think the main advantage of using these plate-based methods is that you can combine them, for example, with fax sorting, which allows you to enrich for a certain population based on some surface marker of the cell that you might know about. It also allows you to combine, for example, with laser microdissection, which can also be very handy if you're trying to search for cells based on their location in your tissue. Just mentioned, you may have heard about DropSeq. The sort of the magic behind DropSeq, the basic idea is that you have these beads, and each bead has a short tag, or a barcode, which allows you to identify it. And then you have your cells, and then you place each cell together with one bead inside a small droplet, so hence the name DropSeq. And then you can carry out the library preparation inside the droplet. And then you can combine all of the droplets together and sequence everything in one lane, or multiple lanes probably. And then because of the barcodes, you can figure out which cell each individual read came from, and hence you can deconvolute the whole process. So that's the clever idea behind that platform. One very active area of research is to compare all these platforms or methods or protocols and figure out sort of which one seems to be not necessarily performing best, but figuring out what their relative strengths are. So I'm not going to go into great detail in this because I think for one of the reasons is because there is currently no consensus in the field which appears to be the best method. There's not even a consensus, I think, exactly on what is the best method of comparing them. And moreover, as I mentioned, there's currently a large amount of protocol development going on. So if you revisit this in about 6 months or a year, there is very likely that there will be sort of a new set of protocols that everyone will be talking about as being the best and most useful one. So that part hasn't really plateaued out yet. So I'm just going to mention two different papers, one from the Enard group and one from the Teifman group, who did different experiments to try to compare this. So the Enard group, what they did was that they took the same cell type, mouse embryonic stem cells, and then they used 5 different protocols to prepare libraries, and they try to sequence them to sort of similar number of cells at a similar sequencing depth, and then they try to compare them in different ways. So here's one of the figures from their paper. So what you can see here is that they set a certain threshold, and then they asked how many genes do we find in each cell for this once we've controlled for the sequencing depth. And as you can see here, the numbers are actually quite different. So if you compare the two most extreme cases here, you see that for the SMART-seq2 protocol, you detect almost twice as many genes as for the DropSeq protocol. So if your goal is to sort of comprehensively find out how many genes are expressed, this might actually be a better bet. Now, of course, there are other aspects that you would like to consider as well, including the cost. So this is a very, very useful and important study to take a look at if you're trying to work out which is going to be the best method for you. So the Teichmann group, they used a slightly different approach. So their method is basically used on looking at synthetic transcripts, or spike-ins that we'll be talking more about later. And what they did was that they used these, so many different experiments had included these synthetic transcripts, which are sort of the same. So that allows you to compare the different experiments, and then they used sort of two different measures, so the accuracy and the sensitivity. We don't need to go into exactly on how they are defined, but here you want to have a high accuracy, and the sensitivity is in terms of the sort of the molecular detection limit, so it tells you how many RNAs did you need to have present from the start in order to be able to detect it. So here, ideally, you want to have something low. And as you can see, there is actually quite a big difference, especially on the sensitivity, between the different protocols. So again, this is important to be aware of when you're designing your experiment. Any questions on the experimental methods? Right, so I think it means that they, I forget exactly what they did, but they were basically asked by reviewers to carry out additional experiments to based on something that was in some of their findings. So I think the sort of the other ones are, they took published data and here's their own experiments, I think. So the sort of, as I said, the starting point for this course is a FASTQ file that you received from your sequencing core or from your sequencing instrument. And the sort of the first step or the first goal is to create what's called an expression matrix or a gene count matrix, or sorry, cell count matrix. And this matrix, the convention that we'll be using is that it's a matrix where each row corresponds to a gene and each cell corresponds to a column. And then the number in each element of the matrix corresponds to suitably normalized level of expression of that gene in that cell. So, of course, some people use the transpose of this for their definition, but that's a fairly trivial operation. And as you can imagine, this matrix, depending on whether or not you have a large number of cells or a small number of cells, it can be either relatively almost square, or it can be sort of long and skinny, or it can have a very large number of columns compared to the number of rows. So you can't really make any assumptions about the overall shape of the matrix just a priori. Anyways, so going back to the first step, which is the reads quality control of the reads. So what we recommend is to use one of the standard tools, for example, FastQC or Kraken. And so here we have been using FastQC, and this is an example taken from experimental data generated by some of our collaborators. So they had 125 base pair reads, and this is one of the summary plots from FastQC. And as you can see here, you have sort of relatively poor quality at the start of the read, relatively poor quality at the end of the read, which is sort of quite common for these experiments. For some reason, there is a drop in quality in the middle of the reads. We don't really know why that is. However, it's very unlikely that this is going to have a major effect on the mapping. So we make a note of this, and we just proceed with this data because we think it's over 100 of the bases seems to have high quality, so that should ensure good alignment. So next step is to align the reads to a reference genome. The traditional way of doing it is basically to take each read and try to figure out where its best match is in the large genome. So this is conceptually straightforward. It's a computationally tricky problem. We like to use, for example, star or top hats. Again, standard aligners. One of the major problems that can occur here is that you have a very large volume of sequencing. If you have thousands or tens of thousands of cells, so this can take a very long time to carry out. And so recently there have been some interesting developments in this field. And these methods called pseudo-aligners. Examples of these are Callisto and Salmon. And they seem to be performing orders of magnitudes faster than traditional aligners with little or no decrease in performance. And we're not going to go into the details on how they work, but they, if you know that you're interested only in the sort of annotated genes, then they are likely to be a very good alternative, and it's likely to save you a lot of CPU time in terms of using them. So
when you try, I mean, is there any of these mappers that consider the whole genome? So if you take, for example, uniquely map.
Yeah, so the traditional mappers do, but not the pseudo-aligners.
Okay, that's a start takes into account the whole genome, and then when you are trying to do the mapping,
yeah,
it doesn't only like towards the transcriptome.
So the pseudo-aligners goes only towards the transcriptome.
But not a star or
No. Yeah, so the question was whether or not the aligners go towards the whole genome or just towards the transcriptome. Of course, what we have to do is to sort of ask ourselves, once we've done the alignment, does it seem to be of good quality, or do we have some reason to be concerned here about the sort of the quality of the reads and the experiment? The answer is that it is very difficult to know in general. Typically, what we have observed in the experiments that we've looked at with human and mouse data is that you will get around 60 to 70% of the reads uniquely mappable. But again, it depends on the protocol, on the read length, and the settings for your aligner. So it's very difficult to give general rules for that, general hard rules. What we do note is that you expect sort of most cells or all of the cells to behave in the same way. So if you find outliers, so cells with for some reason have a much higher or much lower fraction of reads mappable than the sort of the bulk of the cells, then you should probably take a closer look at them and possibly exclude them for further analysis. And I think one of the, at least one of our philosophies and principles when we go through QC is that, in general, we tend to be relatively strict and try to, it's always painful to throw away data because it might be useful, but on the other hand, if the data is poor quality, then you may end up sort of including noise in your analysis, which will make it harder to detect the signal. So it's a difficult trade-off to make, and it's more of an art than a science. Right. So continuing with the example. So this is actually the same experiment that we had before with the FastQC example. So we aligned the reads using star, and then we just count the number of reads for each cell. So this is some 100 cells or something, and they've been sorted. So each bar represents a cell. They've been sorted in terms of the number of reads that they have, and then we split the reads into 3 different categories, whether or not they're unmapped, uniquely mapped, or multi-mappers. And sort of what you can see here is that the guys here with the red arrows seem to have a very low number of uniquely mapped reads compared to the rest. So we basically decided to exclude them. We think that they are likely to be sort of stressed or broken cells that are not good for some reason or another. We noticed that we have 2 funny-looking cells here, where for some reason you have sort of about the median number of reads in total, but for some reason you have about a 500,000 or 1 million or so fewer uniquely mapped reads than what you'd expect compared to how all the other cells are behaving. We have no idea why this is. We basically mark these cells and decide to sort of follow them a little bit more closely to see if there's something else funny going on with them. But in general, here we don't really see a good reason to exclude them just based on this. But it's sort of something to keep an eye on, really. That's all we can say at this point. Next step is mapping QC. So here again, you can use standard tools. And one of the plots that are useful to make is this coverage plot. So what we have here is a metagene. So this is basically we sort of align all of the genes, and then we normalize their length. So here is the start of the gene, and here is the end of the gene. And then we just and then we also normalize the coverage, the number of reads. And we see how what it looks like. So this was supposed to be a full-length protocol. As you can see, the coverage is not as uniform as you would like, but you definitely have a 3' bias. You can see that each line here corresponds to 1 cell. So you see almost all of them behave in a very similar way, except for these 3 guys, which have a somewhat different pattern. We don't know why that is. We take a conservative approach, and we basically exclude these 3 cells from future analysis. So again, that's how we proceed here. Yeah. Sorry, the x-axis. Is it the? So this is the normalized position within the read, within the gene. So 0 means the start of the gene, and 100 means the end of the gene. So I'm looking at all of the genes, but they have different lengths, so I have to rescale them. So basically, essentially, you bin each gene into 100 bins, and then you look at the expression value in each bin, or the number of reads in each bin.
Sorry, I missed what was the upstream methodology for this? Was it Fluidine? Did I miss that bit?
Tallulah, what was the what was the?
This was Fluidine C1.
This is Fluidine, yeah. So Smart-seq 2, right? I think they use.
Smarter, isn't it?
No,
Or did you put your own in?
it was Smart-seq or Smart-seq 2. I don't remember
Yeah.
exactly.
So, yeah, and then finally the next step, the final step in creating the expression matrix is to do reads quantification. So now we can use a standard tool, for example, feature counts or HT-seq, and then we can sort of run from the command line with a annotation file, and then we can produce an output which will sort of contain all this information about the expression of each gene or transcript for individual cells, which is sort of what we were hoping to get. And sort of now we're going to be spending quite a lot of time asking how we can sort of properly do QC and normalization on this expression matrix. So as I mentioned before, we have this feature called unique molecular identifiers, or UMIs, which are essentially a short barcode. Typically, people use 4 to 10 base pairs. Sort of the trade-off here again is that if you use a so there are two trade-offs. One is that if you use a short, the shorter your barcode is, the more of the sort of genomic sequence you will be able to get out to read off, which will sort of improve your mapping. Again, with the read lengths that we have today on sort of 100 plus base pairs, sacrificing 5 or 10 base pairs shouldn't make much of a difference in terms of the mappability. So I don't think that's a big difference. The other reason the other consideration here is that the shorter your, if you have a long barcode, then you increase the number of different combinations, and hence you reduce the chances of collisions, which is a good thing. On the other hand, if you have a long barcode, then you have a greater chance of having sequencing errors in your barcodes, which will lead to different types of errors. So that's a drawback. So again, we need to sort of balance those two aspects, and we'll be talking a little bit more about that later. So I think it's sort of illustrated here in this in the schematic how the barcode is attached. So again, this is typically done in these drop-seq type of experiments where the cells are pooled. So you have 2 barcodes, one which corresponds to the cell, and one which corresponds to the molecule. And what you will do, and what you need to do as you process your reads, is that you need to sort of trim these barcodes off before you do the mapping, and then you need to somehow keep track for each read what its barcodes were as you do the further processing. Yeah. Yeah, exactly. So you have the barcode will typically have 2 components. One part which is unique to the cell, and one part which is unique to the transcript.
Can only be done for transcripts which are known.
Sorry?
So when you put it in the barcode for the transcripts,
Yeah.
will be only for the annotated. Do you know how many roughly transcripts are, or just
So no, so the barcode. So you have these short barcodes which are randomly attached to the transcripts.
And then always enough of them?
No, there's not always enough of them. That depends on how long you make them. If you make them really short, right? If your barcodes, UMIs are only 3 base pairs, for example, right? Then you only have 64 combinations. So then, of course, it's not going to be enough of them. So that's part of the trade-off that you need to figure out. Right. So again, counting these barcodes or the UMIs is straightforward in itself, but there are some issues. As I mentioned, if your barcodes are relatively short, then it means that you may not have enough of them. So basically, 2 different molecules can end up with the same barcode or the same UMI. So that's problem number 1 here. So of course, that should be relatively easy to spot if you look at the rest of the sequence, that if they correspond to sort of vastly different molecules, then you can assume that you have this scenario. Now, a different problem is that you have sort of basically, I mean, yeah, so this can be that you have the different multi-mapping issues where you end up having sort of the same reads coming from different transcripts, but the transcripts look the same because the 3' ends, if that's where you have your tags, are so similar. And finally, you can have this problem where you have errors in your barcodes. So you think that you have more. There's a sequencing error in your barcode, which means that what really was the same molecule, you think are 2 different molecules. So that means that you overestimate the expression of that molecule. So we can try to correct for that in various different ways. For example, if we know or have an estimate of the error rate of the sequencing, then we can sort of, if we find 2 barcodes which appear to come from the same molecule and differ only by a single base, for example, and then when we can calculate the probability that that was the result of a sequencing error, and we can sort of try to compensate for that and try to avoid this problem of overestimating. So again, how to best treat UMIs and sort of use them to get the correct quantification is still an open research problem, and there are methods being developed for this. So one of them, for example, is called UMI tools, which seems to be working quite well. This is something we use, and then you can also try to develop various methods. For example, this one by Green et al., where they use a very simple idea of the sort of collision probability, where they try to calculate or estimate the probability that the sort of multiple molecules would get the same barcode. So we're going to be looking at some data here from an experiment. So here we've plotted each dot in this plot corresponds to a gene, and then plotted here on the x-axis is the number of UMIs, and here's the number of reads. And what we're hoping for here really is this kind of linear relationship that we're seeing here. And ideally, we would like to have it as tight as possible, because that would mean that you have sort of relatively uniformly number of reads per UMI. But this is a kind of result or output that we're expecting from these experiments. So once you process this data, and you can try to get the estimate for the number of unique genes and the number of total molecules per cell, and we can try to plot those against one another here. And here again, you see a sort of a relatively sort of reasonably linear relationship, that if you have more molecules, more unique molecules in your cell, you also observe more different genes. But again, you have a reasonable spread here, which is we think that most of this is biological, but we're not really. We would like to make sure that it's not due to technical issues. Right, so I think we have the first exercise for you here today. So what you need to do is that you need to get some UMI counts and some read counts from an experiment. And this is an experiment that we'll be using a lot in the next few chapters. And then you need to try to figure out what the capture efficiency is and what the variability is. And then you will also need to try to figure out what the amplification rate is for this data set. And I think for all of these exercises, there are answers that you can reveal in a magic way in RStudio. So do you want to explain how to go about with that?
Okay, so you should have RStudio open on every desktop in this class. Is it open, Paul? Or if it's not open, you can just click on the RStudio icon on the left. And sorry, where is it? That's the same. Okay, so you click on RStudio icon on the left, and then if you go to the Files tab here, it will contain all the chapters that we have on the website. So basically, this chapter is number 5. So if you click on it and go to the end of it, you will see the solution to the exercise. It's basically here. And for all the exercises that we have in the next chapters, you can do the same thing. So basically go to the chapter that we are talking about, and then you can find the solution to all the exercises. But better first try to solve it yourself, and then if you can't, go to the exercise and see how you can solve it.